Welcome to our first topic in Form 2 class. This marks our first lesson for the Form 2 work. My name is Teacher Ibrahim, and I hope we are going to learn more as we are going to talk about this topic, transport. Transport in animals and plants entails the movement of materials inside the body of plant or the body of animal. We are going to start by looking at transport in plants. There are some essential elements which needs to be transported within the body of a plant from time to time. They include gases such as carbon dioxide and oxygen. Water is also a basic requirement for the plant because if it is not present in a plant, then the plant may end up dying. There are also some metabolic wastes which need to be eliminated out of the plant body, and therefore they keep on circulating until they are taken to various organs which are meant to store them, or else which are meant to keep them when they will not, since they are not needed within the plant. There are some structures that we are going to measure on, more especially when we'll be looking at the process of transportation of water from the soil to other parts of the plant, because water is a basic requirement upon the process of transportation to take place from time to time. That makes us to find great importance in the root structure of a plant, whereby we are going to look at a certain specimen that I have right here with me. And when you look at this specimen here, this is a dicotyledonous plant. You can see it has got various leaves there. It also has some roots, as you can see. And when you look at these roots, there are some smallest parts that appears to be like the hairs. We refer to them as the root hairs. Root hairs have got some cells which comprise of sap, which is highly concentrated, and these cells are meant to absorb water, and their sap is highly concentrated so as to increase the osmotic pressure to allow more water to be absorbed from the soil into the plant through the process of osmosis. There are two types of sections that are obtained when the specimens are being dissected in a biology lab. The first one is referred to as a longitudinal section, while the second one is referred to as a cross section. The two types of sections, as I said, are transverse section and longitudinal section. If we dissect this root from up to bottom, or you can call it vertically, that one is referred to as the longitudinal section. If we cut it horizontally, or from left to right or right to left, then that one is referred to as transverse section. Here is the diagram which comprises of the different types of sections that we can obtain when dissecting the plant specimen. This one is an example of plant specimen that have been drawn on this page over here, and we are so much interested in the root region. That is why it has been circled, as you can see down here. If this part was cut vertically from top to bottom, then we are going to obtain a structure that looks like this. Therefore, it is referred to as longitudinal section, from the word longitude. When you look at the longitudinal section of root, there are various parts that you are able to see here. In the first place, you are able to see the root hairs. I told you that they are the hair-like structures which are found on the root. Apart from the root hairs, you are able to see a certain central tubular structure in it, which is referred to as the xylem. Function of the xylem is to transport water and mineral salts from the soil into the plant body. We also have the xylem vessel over here, as you can see. The xylem vessels transport manufactured food from the leaves of the plant to other parts of the plant, such as storage organs and maybe the young developing birds which may require such food. The phloem is responsible for transporting food to them. When you look at xylem and phloem, the major difference that you can note is the xylem is found at the centermost part of the root, while the phloem is found just besides. And the phloem are more because more food is always synthesized by each leaf from time to time, and they are transported. We also have another part within the root which is referred to as apical meristem. The apical meristem, this space inside here, as you can see, comprise of young and actively dividing cells which keep on dividing from time to time, and above them there is the root cape. The root cape is just an impermeable part of the root which is found at the root tip, and the function of the root cape is to protect the apical meristem, which comprise of the young actively dividing cells, as the root pushes through the soil particles. Therefore, the root cape only offers protective function, while the apical meristem is the one that comprises of the actively dividing cells. All these three can be divided into three major regions. We have the first region referred to as region of cell differentiation. The region of cell differentiation comprises of structures which are fully developed and they are mature, while there is the lower part which comprises of cells which are yet to mature, but they have not yet differentiated, but they are just about to differentiate. They are referred to, that region is referred to as the region of cell elongation. In the region of cell elongation, the cells tend to grow longer, and therefore we also have another region just beneath it referred to as the region of cell division. The region of cell division comprises of the apical meristem and the root cape. Therefore, in this region, there is the highest rate of cell division taking place, which results into elongation of the entire root. During this process, when these cells are dividing within the apical meristem, they will end up being many, and when they increase in number, they end up occupying a larger space. A large space being occupied will force the root, will push the root cape away from the apical meristem, because all these cells require to occupy that space, and in that process there is an increase in the size of the root, and that is how the process of growth take place in plants. And the same always take place in the stem as well, whereby the root tip increases in size due to the process of cell division. When the process of cell division takes place, then it causes the root or the shoot to increase in size, hence there is an elongation. I would like us to look at this structure of the root hair cell. Remember, the root hairs are these small hairy structures that we saw up here, above here, I think you can remember. But right now, I would like us to look at the structure of the root hair cell. I said it earlier on that inside the root hairs, there are some cells which are referred to as the root hair cells. When you look at the structure of the root hair cell, it is slightly different from the other basic cells that are found, the basic structure of cells that are found in plants. There is a small projection here that is referred to as the root hair region. And the other parts of the cell just remains to be similar to the other basic structure of the plant cell. Just like the other basic structure of the plant cell, the root hair cell comprises of the cell wall. It also comprises of the cell membrane, the cytoplasm, cell vacuole, and the nucleus as well. There are some few adaptations that the root hair cell comprise of, which enables it to be absorbing more water from the soil. Those adaptations include, it has a highly concentrated sap which enables it to absorb water from the soil. Therefore, we say that the highly concentrated sap increases osmotic pressure, increases surface area for absorption. The root hair cells are also numerous in number. Even though the root hairs have a short lifespan, they are continuously replaced for efficiency of water absorption. Another adaptation of the root hair cell is they are thin-walled to facilitate the process of absorption of water, and therefore that marks the end of that subtopic under the root hair cell. We also move to the internal structure of the roots and the root hair. As you can see on the board here, we are having some different types of root structures, that is the dicot root and the monocot root. I drew them together so that we are able to compare the two and we can be able to see how different they are from one another. When you look at the dicot root, it comprises of the root hair just like the monocot root, and it also comprises of the epidermis. Epidermis is the outermost region of the root, and its role is to protect the inner part of the root from mechanical damage or from microbial invasion. We also have the cortex, and the function of the cortex is to store water and manufactured food. The cortex comprises of parenchyma tissue. It also comprises of the collenchyma tissue as well as sclerenchyma tissues. All those tissues that are found within the cortex, the first two that I mentioned, that is collenchyma and sclerenchyma, are strengthening tissues. Collenchyma is a tissue which is thickened by pectin and cellulose, hence making it to serve as a strengthening tissue within the cortex. While for the case of sclerenchyma tissue, it comprises of a compound known as lignin. Therefore, we say that sclerenchyma tissue is lignified, and that also prevents it, that also enables it to act as a strengthening tissue. The only difference between collenchyma and sclerenchyma is collenchyma tissue, as much as they are both found within the cortex, collenchyma tissue comprises of cellulose and pectin as their strengthening tissue, while sclerenchyma comprises of lignin. Then we say that sclerenchyma tissue is lignified. When you look at these two, the other difference that you will be able to note is the presence of the vascular bundles, that is xylem and phloem. The vascular bundles, for the case of the dicot root, it is star-shaped, while for the case of a monocot root, the vascular bundles are just spherical in shape, and they are just found to be surrounding the pith tissue, which is found at the center of the root. They both contain endodermis and pericycle, which are the single layer of cells. This one here is a layer of cell, and this one is also another layer of cell, and they protect the inner part of the tissue. We also have the stem of the dicot plant and the monocot plant, and when you look at this stem of the dicot plant, we have the vascular bundles, that is xylem and phloem, which are located on a structure called the cambium. In some cases, we can call them the intervascular cambium because they are found between the vascular bundles. This intervascular cambium, its role is only to suspend the vascular bundles and keep them into position, while the epidermis protects the inner part of the plant from mechanical damage or from microbial invasion. On the other hand, when you look at the monocot stem, the arrangement of the vascular bundle, that is xylem and phloem, may be different in sense that when you look at the dicot stem, they are regularly arranged in the cambium ring, while for the monocot they are not. In a monocot they are scattered within the stem. I would also like us to move to something else, whereby we are going to look at the process of absorption of water and mineral salts, which occurs from the roots of a plant to the other parts of the plant. We have a diagram of a tree here. You can look at its roots. There is a certain part that I have just circled here, as you can see it, so as to make us concentrate only on this part. If this part was cut in a transverse section, then another part, a small part, is chopped out of it. This is how it would look like. There are some root hairs here which enables absorption of water, and that is why I've written here water entering into the root, that is absorption. Since there is a higher concentration within the root hair cells, there is an osmotic pressure which will build up inside the vacuoles of those root hairs, hence making them to absorb water in the process of osmosis. Once the water has been absorbed, it will reach the inner part of the plant, that is the xylem vessel, which enables transportation of that water from the root to other parts of the plant. I would like to describe the process of absorption of water. When there is water in the soil, the root hairs may absorb the water in the process of osmosis. When the root hair cells absorb the water in the process of osmosis, the content within the subvacuole become dilute. Hence, we refer to it as a hypotonic solution. When it becomes dilute, or when the concentration lowers, remember the other subsequent neighboring cells are still highly concentrated than the root hair cell. This makes the water to be absorbed from the root hair cell to the neighboring cells which are found within the cortex. When the when the cells which are found within the cortex have absorbed the water in the process of osmosis, they will also become lowly concentrated, and that makes the water to be absorbed by the cells which are found within the endodermis, because in the endodermis the concentration will still be higher than the concentration of cells which are found within the cortex region. When the water has been absorbed to the cells which are found in the endodermis, the cells within the endodermis region become lowly concentrated, or they become hypotonic, and when these cells within the endodermis become hypotonic, then the pericycle cells will absorb the water from the endodermis region in the process of osmosis. This process will continue until the water is availed to the xylem vessel. And when the water has been availed to the xylem vessel, it will be moving from the xylem to other parts of the plant until it reaches the stem, the leaves, so that it can be used in various processes. Therefore, that makes us to reach something which is another process which is called transpiration. In the process of transpiration, remember, for this water to move from the xylem to other parts of the plant, there are 4 major forces that facilitate movement of water. And before we go to the next one, I would just like to mention those 4 forces. They are transpiration pull, capillarity, cohesion and adhesion, or you can call it cohesive and adhesive forces, and we also have another one which is referred to as root pressure. But we'll come to talk about those ones when we'll be talking about the process of transpiration as a topic. But all in all, please don't forget about that. Let's move to the next part. We are going to talk about the structure of the xylem tissue. Remember, xylem tissues are very important because they are the ones that aids in transportation of water and mineral salt throughout the plant. There are four types of thickening in the xylem vessels, namely annular thickening, that is thickening whereby lignin is present on the walls of the xylem vessel, and that lignin prevent the xylem vessel from falling in case there is no water. And we have the type of thickening which is called annular thickening, reticulate thickening. You can see how the structure looks like, and the bordered pits, we have some small amounts of lignin which are found on different parts of it. And lastly, we have spiral thickening. Spiral thickening is whereby the lignin is found on the walls of the xylem vessel in a kind of shape which is referred to as spiral shape. We also have another type of xylem vessel which is referred to as tracheids. I said initially that the xylem tissues are divided into two, the xylem vessels and the tracheids. The xylem vessels, I've talked about how they are lignified, the different forms of thickening, that is lignification process. And for now, I'm talking about the tracheids. Tracheids are the types of xylem tissues which have some tapering ends. You can look at these ends here. I refer to them as tapering ends. And when you look at them, they have pitted walls. They have all their cross walls have been pitted. These ones are small holes that I'm talking about. And you can also look at this one. It has a lignified wall which are strengthened with bars. You can see these bars over here which makes it stronger. Tracheids are always considered very efficient when it comes to transportation of water and mineral salt compared to xylem because they have pitted walls throughout their cross wall. They have pitted cross walls all over. That is when it will be transporting the water. When the water reaches this small hole, it can get to all the other parts of the plant compared to the xylem vessel, into which the water needs to move along the xylem vessel until it reaches the exit point, is when it gets to the other parts of the plant. I have a wall chart here comprising of the setup of how a certain apparatus, which is referred to as a potometer, may be used up. Potometer is used to measure the rate of transpiration. Transpiration may be taking place in all the plants, but in different rates. And the reason as to why it is taking place in different rates, because different types of plants have got different adaptive characteristics depending on where they originate from, depending on the types of soils onto which they grow, among other factors. Therefore, these apparatus here, potometer, can enable us to know the rate at which transpiration is taking place within a plant. Let's look at it and see which part plays which role during the time when we are checking on the rate of transpiration. It comprises of a leafy shoot which must be placed on top of it, and that leafy shoot is able to absorb water, and the water is placed in a delivery tube in the sense that it is joined to a capillary tube from the lower side. This capillary tube is immersed into a container, into a beaker comprising of water. This is to ensure that as much as the leaf is sucking some water and it is lost into the atmosphere, the water does not get finished up in the beaker. During this process, we also need to have a water reservoir. As you can see here, the water reservoir ensures that the water level doesn't go down, because in case the water is being lost to the atmosphere, the water level in the beaker may drop. In case of such a drop, the technician who is carrying out the process can open up the tap so that the water can get from the reservoir into the beaker. That ensures a continuous flow of water within the entire system. We also have a bubble here. I think you are able to see a small bubble here. It is so small. Let me try to point at it. There is a small bubble here inside the capillary tube, and the function of this bubble is to enable us know how much water has been absorbed by the plant, or else how much water has been eliminated out of the plant, because this bubble will tend to move towards the upper part. We also have a ruler mounted close to that bubble. I think you can see a ruler here. This one is a ruler which shows some measurements there, and we'll be able to know how far the bubble moved within the capillary tube, and therefore we can use that to calculate the rate at which transpiration took place. For example, if it was able to move maybe for 5 centimeters, then we can now put that 5 centimeters over the total length, maybe 30 centimeters times 100%. That enables us to know the rate at which water moved. We also have a retort stand here, but its major role is to ensure that there is steadiness for the entire setup, so that it just holds onto the capillary tube, which comprise of the entire system. We have also looked at the xylem structure, which is one part of the vascular bundle, but I would like us to look at the phloem structure, which is also part of the vascular bundle. I said earlier that phloem structure, as you can see it over here, it comprises of some regions which enables it to which enables transportation of manufactured food. As you can see it comprises of the plasmodesmata, which connect sieve tubes with the companion cells, and it also have various parts like the prominent nucleus. I think you're able to see this prominent nucleus over here, and I think you understand the role of the nucleus, that is controlling all activities that are taking place within a cell. Remember, this tissue, its major role is transportation of manufactured food. We have some parts here which facilitate transportation of manufactured food. As you can see, these ones here that we call the sieve tubes. The sieve tubes facilitate transportation of manufactured food, whereby the food is manufactured and it gets into some sieve tubes, and it is transported all over the plant body. Remember, this food is transported to storage organs, and it may also be transported to other parts which may just be requiring some food. For example, parts such as helping parts and other parts such as the storage organs, as I said earlier. That marks the end of our subtopic for today, that is transport in plants, and I'm yet to cover the second topic, that is transport in animals. Thank you so much for watching the video, and I hope that you have learned something, and next time you will also learn more, because I'm still yet to cover some more. Please don't forget to like, subscribe, and if possible, press that notification button so that in case next time when I'm going to upload another video, you will be with me and we are going to learn. Thank you so much.